Hi guys, so I'm Audrey,
And I'm Kyla.
and this is our second presentation on the hydroxyl group and BH4 interactions and the mechanism of the human enzyme phenylalanine hydroxylase. So first, we're just going to talk a little bit about congenital disorders and what they are. So 1 in 33 babies each year are born with a birth defect. And a lot of birth defects can be caused by environmental factors such as smoking or alcohol during pregnancy, but there are certain congenital disorders that can be inherited through genetics. And many of these, like the one we're going to talk about today, are due to deficiencies of certain proteins that we need in our bodies. And another just small fact about them is that they can range from being barely noticeable to being fatal to a child at birth. And some prevalent examples in our society include Down syndrome and cystic fibrosis. So the one congenital disorder we're going to focus on is phenylketonuria, or PKU for short. So 1 in 10,000 newborns are born with this disease, and it's caused by a deficiency of the enzyme we're going to talk about, phenylalanine hydroxylase, which converts phenylalanine to tyrosine. The issue with this disorder is that there are no existing supplements for PAH, so sufferers of PKU have to limit their intake of phenylalanine. And this can be kind of hard in our society because not only is phenylalanine found in meat and fish and basically anything with protein, it's also found in artificial sweeteners. And this picture here shows a Diet Coke bottle, which has a warning label for phenylalanine. And the only existing cure for PKU is really just to maintain a diet free of phenylalanine, which can be really hard.
PKU is also known as Falling's disease, and the inheritance of PKU is autosomal recessive. And what that means is basically both parents have to be carriers, and and in order for their baby to express the disease, they pass on both recessive alleles to their child. Um, and the product of this reaction that we're going to be talking about is tyrosine, like Audrey said. And what makes tyrosine so important is that it's a precursor for the biosynthesis of certain neurotransmitters. So without sufficient enough amounts of the tyrosine, it can cause cognitive disabilities, and as well as the buildup of phenylalanine can be toxic to the body. The phenotypic expression typically of PKU can result into usually lighter hair color, and if there is a buildup of phenylalanine in the body, it can cause a certain type of like musty smell within the carrier as well. And along with that, PKU is tested 1 to 2 days in a newborn after they're being born, and this further emphasizes the importance of the disease because it is revealed very quickly within their lives. And the disease can cause complications for the mother in her decision to continue breastfeeding or to play it safe with the phenylalanine-free formula. And it's obvious that the mother can't really determine the phenylalanine levels in her breast milk. However, a lot of other nutritious benefits can come from breastfeeding, and so it causes kind of a complication in the mother's decision to choose whether to breastfeed or to be 100% safe.
So here's our thesis. The enzyme phenylalanine hydroxylase, which with its cofactor BH4, catalyzes the reaction to convert L-phenylalanine to tyrosine. The enzyme contains binding sites for L-phenylalanine and BH4 through the two specific amino acids, glutamate and histidine. These charged residues create a stable environment for L-phenylalanine to be converted into tyrosine. The deficiency of this enzyme causes an autoimmune disorder called phenylketonuria, which can cause mental disorders and seizures because of the lack of tyrosine in the brain.
So for the general overview of what our biomolecule PAH does is it catalyzes the hydroxylation of L-phenylalanine into L-tyrosine, and as we move on throughout our presentation, we can then go towards a more deeper analysis of the mechanism as more of the interacting biomolecules are introduced.
So here's a picture of the three different domains of PAH. It is a dimer of tetramers. So each dimer contains two catalytic domains, a regulatory domain, and the C-terminus oligomerization domain. So as you can see in this picture, there's a little orange sphere, and that represents where the catalytic domain is going to be because that is the iron atom. So the catalytic domain contains the active site where there are binding sites for the cofactor and the substrate. And then the green oligomerization domain connects the two subunits in this protein.
Here is just the monomeric form of the structure, and it's just useful for being able to visualize the allosteric regulation of PAH. And so phenylalanine itself allosterically regulates PAH. Normally, the enzyme resides in two forms, the resting form and the active form. In the resting form, or the T state, the N-terminus residues cover the active site. And in order for the enzyme to move into an active form, L-phenylalanine must bind to this regulatory domain. So just to go back to this monomeric visual, the regulatory domain is near the N residues. And so moving forward to this cartoon in the multiple monomeric structures all together interacting, the regulatory domain is binded to phenylalanine, and that causes a conformational change, which will then move these N-terminus residues away from the catalytic site, and that allows for the catalytic domain to be exposed to phenylalanine in that site, and that's where the hydroxylation does occur. To help visualize this further are these two visuals. And so, like I said, the resting state and the active state exist in an equilibrium. And so when they're binded to that certain active domain dimer, that's what has caused this dimer. And here in this visual, they're at resting form and covering the catalytic site. But as it conformationally changes, they then move away from the N-terminus, and the active site can then be exposed.
Okay, so next we're going to talk about the two amino acids that really play a role in PAH. So the first one is histidine. So as we know, histidine is positively charged and aromatic. So that allows it to interact with the cofactor and L-phenylalanine because its positive charge interacts with the hydrogens on BH4, and the aromatic structure allows it to interact with L-phenylalanine, which is also aromatic. So because it has two specific structures, there are two really important residues, histidine 290 and histidine 285, and they each interact with BH4 and L-phenylalanine respectively. And the other important thing about these two histidine residues is that they bind directly to the iron atom contained in the active site. And iron is also a super important part of this enzyme because it has multiple oxidation states, which we're going to see contributes to the function of this enzyme later on. And the other important amino acid we're going to talk about is glutamate. So the structure of glutamate allows it to be deprotonated and have a negative charge on its R group. So there's one glutamate residue which really plays a role, and it's glutamate 330, and it's oriented such that BH4 can bind really close to it. And because it has a negative charge and BH4 has a lot of positively charged regions, it forms a stable environment for BH4 to bind really tightly to the enzyme. So now we're going to talk a little bit about the cofactor for this reaction. BH4, which is short for tetrahydrobiopterin, is a really important cofactor in many neurotransmitter reactions. So it has multiple regions with hydrogen atoms, especially as you can see on this picture, some amine groups and some alcohol groups. So it's able to donate oxygens and hydrogens easily and work together with other compounds such as oxygen and water to replace these atoms on its structure. It's also able to supply electrons to reactions because it has some double bonds, and it works in our reaction that we're going to talk about. It works well with oxygen, and then it becomes hydroxylated to 4A-OH BH4 and then can go right back to BH4 by some more redox reactions. So as we said before, BH4 is the cofactor for this enzyme, and lots of enzymes need cofactors in order to function completely. So here's a little bit more detailed picture of the structure of BH4. So how it works in this reaction is that it supplies electrons from its double bonds, and it works together with O2 to add the OH group to phenylalanine to convert it to tyrosine. And it's also hydroxylated itself by O2, and then it becomes 4A-OH BH4, and that can be recycled back to become BH4 to act again.
So with that knowledge, we can combine the phenylalanine in the reaction with its cofactor BH4, and the incorporation of an oxygen molecule on both the cofactor and subsequently the substrate are hydroxylated, as can be seen here with this L-tyrosine product, and like Audrey said earlier, the 4A-OH-BH4 intermediate. But to fully understand how the enzyme is able to do this, it can be looked at within the active site. And so, like she said earlier, that this is just a blown up picture of the catalytic domain in the enzyme. And like she said earlier, the orange dot represents an iron atom, which closely associates with these two histidine residues, as well as this glutamate residue. And the iron itself, in conjunction with these amino acid residues, helps to mediate the incorporation of one atom of oxygen into each L-site phenylalanine and BH4. And so when we want to look up at a close-up of the mechanism, it can be seen here that this is BH4, this is the L-phenylalanine, and this is the residues in the enzyme. As the oxygen molecule is incorporated, iron helps to act as a transition metal. And because it has many oxidation states, it can allow for the transfer of electrons, which follow the addition of oxygen within these substrates towards this reaction. And so what can be seen here is that the BH4 cofactor, which is this molecule right here, is oxidized first as a bond is created between the cofactor and the iron. And then as that bond is cleaved, the 4AOH intermediate is then formed as it's hydroxylated, and the other oxygen atom remains on the iron, and its oxidation state has changed. And so continuing on from there, this allows for the phenylalanine to then attack the remaining oxygen on the iron, and the incorporation of that second oxygen, as well as the hydrogen that will then follow, helps to create the product of L-tyrosine. And so this flow of electrons is paired with the movement of oxygen, as can be seen in the changed oxidation state from 2 to 4 in the iron. And the amino acids surrounding the iron help for stabilizing the reaction itself. Both histidines and glutamate can be easily protonated and deprotonated, and this helps play an important role in stabilizing the environment for the reaction. And along with the L-tyrosine, the intermediate 4AOH-BH4 is formed. And so in order to keep the reaction running again, the 4AOH-BH4 must then dissociate from PAH and become regenerated to BH4. And that regeneration helps to just further complete the mechanism, and all of the products are formed and all of the cofactors are reformed. And so how that happens is it is dehydrated and reduced off of the enzyme, and from that a water molecule is lost and the BH4 is then reformed. So in conclusion, there are many steps that regulated the function of PAH, like we have seen throughout the presentation, and that those steps can be allosteric binding, which then would cause a movement of the N-terminus, and also not just the actual catalyzation of the reaction itself, but even just the regeneration of the cofactor. And all of those things can cause complications for catalyzing phenylalanine into tyrosine. And so that's why the treatment for PKU so far has just been best handled from its exclusion from the diet, because it can be difficult to test exactly where and what a mutation on this enzyme is causing. So that's it. Thank you.
Thank you.